Very quick review. We were talking about histone modifications, and you need to be able to explain what they are, recognize them, and talk about the effect, whether it's going to be like the effect relatively on DNA structure. You can talk about the effect on transcription, translation in a roundabout way, things like that as well. And so this is just a picture that came from, I forgot which review, just to show you the different modifications that can occur. And we went over ubiquitination, which is also called ubiquitinylation. And that's a small protein that gets added on towards the terminus. And it had two roles. One is many times it's involved in marking that the protein needs to be degraded by proteases. That's not what's happening here for histones. And the secondly is the fact that it helps increase transcription by affecting the affinity of the histone for the DNA. Okay, sumo protein is very similar to ubiquitin, and so it behaves similarly as well. And we talked about acetylation, and that the R group in this instance here would be the lysine. So the lysine goes from being positively charged oftentimes to when it's neutral. So then you need to be able to explain the effect, whether that would increase or decrease transcription. Okay. And likewise, a lot of these histone modifications are involved in oncogenesis and tumorigenesis as well. Okay. And we talk about methylation and phosphorylation. Methylation is a little different because if it's a lysine, it can either be monomethylated, dimethylated, or even trimethylated. If it's trimethylated, it's got a permanent positive charge. I shouldn't say permanent because it can be demethylated as well, but it's got a positively charged. It's just really big and bulky. If it's monomethylated or dimethylated, it's neutral. So it's kind of like a fine-tuning way of doing things. Whereas phosphoserine, we already talked about it, and the serine goes from being neutral to having a phosphate group on it. And what's the charge on a phosphate group? Negative. And so you'd have to then think about and use the logic that's behind biochemistry and talk about what that would affect. That would have on DNA binding to the histones, or you could say the histone's affinity for DNA, and ultimately whether that would increase or decrease transcription. This is just the same picture that I showed before, kind of showing this histone, I don't know, metabolome, whatever, transcriptome, whatever the biologists would like to call it, the language of histone modification. And then in particular, the example that this one was showing was histone deacetylase and histone acetyltransferases, which are antagonistic enzymes in the acetylation process. And they are very specific. They're highly specific for the lysine and also for the histone itself, which histone they can modify. Okay. This should be a review, I would think. I would assume you would cover in genetics and or cell molecular. But there are a couple ways of monitoring nucleic acid structure. One of them is this way. Do we know what this is? They're using UV-Vis, right? And the reason why is that nucleic acids, RNA and DNA, both absorb light around 260 nanometers, okay? Whereas tyrosine, tryptophan, those are the two biggies, in proteins absorb light closer to 280 nanometers. But what are they doing to the DNA? What are they literally doing to it? That's UV-Vis. What's this? What? Yes, they're melting it. It's called a thermal melt. And so literally you add temperature and you measure the UV-Vis because it goes from whenever it's highly structured, typically it has a lower absorption, where if it's just randomly loose in the solution, then the absorption will increase. And this inflection point is called the TM. And TM is a unit. If it's got more GC content, the TM will be higher. Obviously, if it's a really long piece of DNA or RNA, then it's going to be higher. If it's primarily AAT or AU, then it's going to be a little bit lower than GC because it's the number of hydrogen bonds. Assuming it's all canonical, RNA is a little bit more troublesome, or I shouldn't say troublesome, a little bit more specific. Okay. All right. And this brings us back to this idea of RNA structure, and it is more complex than DNA. And some of this is not, I actually haven't checked the most recent. If you got the most recent edition of the book, they may have updated it, but it used to not be in the book. The jargon's used for nucleic acid structure. So, I thought the same so many times. Yeah, it still works. Okay, it's all good. So why is RNA structure more complex than DNA structure? Why?
Be modified?
It can be modified. That can help increase its complexity. What else? DNA can also be modified. Bacteria use it for their, I call it their immune system. I'm sure that microbiologists kind of cringe at that. They actually do like methyltransferases and stuff to DNA. What else? Why else is RNA going to be more complex than DNA? There are different types of RNA. What else? You kind of zoom in on different rules. What is the difference between DNA and RNA? That extra alcohol, and that extra alcohol can do a world of difference, right? Because it's not only nucleophile, as we mentioned before, but they also do hydrogen bonding. And it can also chelate to metal ions and stuff where hydrogen cannot. So you need to know the difference between DNA and RNA, and then be able to talk about factors that affect the RNA structure, which I don't think I actually have it on there. I don't. So let's go over that. We've gone over the differences between the two, but what are some of the factors that can affect RNA structure? I thought I had put it on in another slide, but I didn't. Just in general, what's one factor that's going to affect the structure of RNA? Some of the protein or DNA doesn't matter. What's just one factor? Think about it. Quentin, what's one thing that can affect the structure of this?
pH.
Exactly. pH is a biggie. In fact, some of them will literally change conformations based off of pH. And what else can affect it? Temperature. I thought we just did this with DNA. Temperature is a big one. You increase the temperature, you're going to increase the fluidity in the movement. Okay. What's another one? It's actually kind of similar to pH. It can even perturb pH. It's probably one that you guys.
Salinity?
Yes. Yeah. There's another term that they use, but it is salinity. You know, it's ionic strength. Okay. Because that way it covers more than just salt. And with ionic strength, one thing that's a little different that you guys probably don't know that I was hinting at is metal identity. So RNA and DNA too, they can both bind because they have phosphates, and RNA can have the alcohol. They can bind metals, and based off of which metal ion that it's bound can affect its structure. Okay. pH, temperature, ionic strength. One other one is just concentration, right? If it's dilute versus if it's highly concentrated. And then some others. And then any binding partners. Okay, sometimes they bind, like for example, the ribosomal RNA, its structure changes by being bound to the ribosomal proteins. Okay, so if it's, I don't know how to say that, like ligands, I'll just put it all together. Ligands, cofactors, or coenzymes. They can help dictate their structure. Okay. All right, so now we're going to talk a little bit about structure. Oh, yay. First up, this can happen to a lower extent in DNA. RNA can form very, very complex loops. You know, you can see the normal helix that you're used to seeing in DNA and RNA. RNA can also do that. they can form what's called a hairpin loop. And the most common is that tetra loop that we talked about earlier. So there's a tetra loop is the most common, which just means there's 4 nucleotides, but they don't have to be. There are also ones that have 6 and 5 and things like that. I don't know that you can ever have one less than 4 because of steric strain in the bonding. You can have a bulge where only one side has the loop. It doesn't actually have to be both sides. You have both interior ones where it's not on the end. And then you can have like this junction within RNA where multiple helices all come together. Okay? Oh, whoops. I didn't include it in the thing. So the broad strokes here when it comes to structures, there are 3 main aspects. They have loops. They have helices. Okay. And they have what they call joining regions. Okay. And the reason why I put this on here is when you start to look at the secondary and tertiary structure of RNA, they use very distinctive. It's a different way of numbering and talking about things than what you've seen with proteins or DNA. The loops, a lot of times they will abbreviate as an L. The helices, they abbreviate as a P, which is for paired region, which is kind of hokey because it's a helix. And joining is a J. Okay. And literally, a joining region means that it's a section of RNA that connects 2 helices. Now, I'll show you in a moment what I mean. So let's look at an example. This is a hepatitis delta virus, HDV. On the left would be like the proposed secondary. It's one way to show the secondary and tertiary structure, but on the right is the actual crystal structure. What they've done is they're just showing more like a ribbon versus trying, because it's very kinky, as you can see here. Okay. But if you notice on the little way that they write out the sequence in the structure, they use these P's and J's, and sometimes they even use L's. This one has 1, real one. But I don't know if I put in 1 for RNase P or not. But RNase P protein, for example, is huge, and so they come about L5 and L this.
So we're talking about not just human RNA structures, we're talking about all RNA structures. Because in human RNA structures make those like helices and stuff like that.
They make all this kind of stuff too.
They make so like they fold on each other. The RNA folds on each other.
It can.
So how does it get translated if it's like all folded on itself?
Because it depends on the type of RNA. What kind of RNA gets translated?
mRNA.
It's mRNA.
So mRNA doesn't fold on itself.
It can. There can be structures to it, but then what would happen is that the ribosome will help unwind it.
Okay.
You know, you can imagine it being threaded through, and you know, you can have different coils and stuff. Structure can make it more difficult, but it's not. This is nice little long piece of line, right? And the tRNA has a very distinctive shape that all tRNAs have. Okay, the basic shape. This one is a ribozyme. So let's follow along. In the beginning, let's see, 1, 2, 3, 4, 5. Okay, so this is the 5 prime. It always goes 5 prime to 3 prime. And see it here. And if you notice the 5 prime, oh no, I was wrong. 5. Where's the? I'm going to try to count. Right, 5, 4, 3, 2, 1. No, I was going the wrong direction. Here it is. Okay, it's kind of the wrong way. This is the 5 prime end, going 5 prime to 4 prime. 5 prime to 3 prime. I cannot talk today. I apologize. One thing about it is that's different than a protein. You know, when we had proteins, the helices were alpha helices. They were all next to each other, you know, in the primary sequence. In RNA, even though it's a single transcript, the parts of the helix can be very, very far from each other in the primary sequence. It has a primary, secondary, tertiary sequence, and quaternary sometimes sequence, just like a structure, just like a protein. But if we look here, this is the 5', and then it bends over, goes down this way, it goes over here, you go on over, la la la, la la la, up, and there's the 3' end. Okay, so what we can see here is that the very first helix, if you start with the 5' end, gets this structure of P1. Okay, then the next helix would be P2. So any of the nucleotides that's between helix P1 and P2 would be called J12. Then we go down here, and it may very well be that the third helix, which is this little one, and then we have this little piece of helix that sometimes they give them really weird names. This one's a never, and there are two ways that they can. One, sometimes they give them a decimal name because it's one that's not conserved throughout the species. Just like sometimes there'd be one that's missing. I wish I would have included tRNA. Because all of a sudden there wouldn't maybe just not P13. Like what happened to P13? Well, it's because in E. coli it's there, but not in Bacillus or whatever. And so they just they based it based off of their structure. But this one of the reasons why is because in the three-dimensional structure, if you see, it's only two nucleotide base pairs. And so it forms like a bigger helix, a part of it. Sort of like, there's part of the helix that means it, and it's kind of right there. But we can see in this instance here that the part that joins helix 1.1 to helix 4, P4. And so these nucleotides right here are in this joining 1.1-4. And this always goes 5' to 3'. That's why this one goes 4 to 2, because when we go here, this is helix 4, then it joins up to helix 2. And so these literally means it's on the 5' side 4, and then the 3' side of it would be 2, helix 2. It's not on here, but the way that they would do it, usually in papers that talk about RNA, this one only has one loop, but they would call it, oh, whoops, I'm sorry, I thought I was writing. They would call this loop 4, because it's the loop that's at the end of helix P4. By the way, that's the UNA binding domain, which is another type of RNA. So they're very, very complex. And so all of this forms this in vivo. And so literally if you start off, it's kind of hard to tell, but there's the helix here, and it goes down, because the helices themselves are not straight. They can be kinked as well, and so on and so forth. This would be the loop region. Then we can get some really cool, this one's really cool. This one's called the kissing complex. So kissing complex, it's kind of hard to tell, but what they're showing is the part that looks like my fingers, which on the video, you can't see it. That little part right there, you see those are two helices loops at the end. So this is the helix, that's the helix, those are the two loops, and the loops are not perfectly straight. So they actually kind of form like a little latch, almost like a little Velcro or something. And what's really interesting about this structure is they found out that in using biophysics that it's got different tensile strength. And so if you take it and you just barely start to add pressure for it, it's like silly putty, it stretches. Whereas if you add a lot of pressure, it snaps. So, but yes, it's actually got quite a bit of strength to it. That's called a kissing complex. Oh, I did include, okay, good. This is the one that I worked on. This is the secondary structure of the tRNA. At the time, they didn't have a crystal structure. Okay, so just I want to point one thing out. This part right here, this what is this? Is this green or blue? Whatever this color is, that's not naturally there. Those were nucleotides that we added on in order to make a circular construct. Okay, so what we can see here, this is helix P1. No, this is the 5' end. Helix P1, P2, P3. Helix P4 is this section to this section. So that's why if we were naming these, or we are naming it, this region, this is a joining region right here. So we know it's going to be a J. what number comes first? What helix is it joining on from the 5' end? So you have to look at the numbering. Remember, the 5' end will have the lower numbering, so it's going down this way. That's P2. This is P19. P19. Now it's P4. And then that's the 3' end. So which one was on the 5' end? Is it 4 or 19? It's on the 5' end of that joining region. No, it's 19. See, this is the 3' end. This is the 5' end. So that means this part up here is 5'. This part here is 3'. So if we want to, we can actually trace it. We start right here. We go up. We go around. I did a lot of work on helix P3. We did have helix P4, which I can explain what that is later on. Then we go, and this one's called 5.1 because it looks a little different between the different types of bacteria. That's just a bulge. And then there is no 6, and this is Bacillus subtilis. There is no 6. The 6 was not evolutionarily conserved. But we have a little bit of 7, 8, 9, 10. 11 is ridiculously tiny. 12. And then it jumps all the way to 15. 15.1, and that's because Bacillus has an extra one. 18, 19, and then back to 4, 1. So that's why this one literally means that this section right here is joining 19 to 4. This next section, this one up here, let me try and find another one that's not. Like, I guess in theory you could call this a joining section. And on this end, it'd be joining 12 to 11. Because it's joining, here's 12, there's the back set of 11. Okay.
So the other side of that circle would be 11 to 12.
Exactly. Exactly. Good eye, good eye, good eye. And I talked about the loops. This one would be called L8 because it's the loop that's on the end of helix 8. That would be L5. This one right here would be L5.1. It's just too small for me to write in it. This would be L15.1, so on and so forth. So that's just the way that they do the numbering. In case you're wondering what this was for, that's the proposed catalytic active site for this enzyme. Because the RNA is the catalytically. It's the part that does the enzyme, the activity. And so that's what this is from one of our papers. Okay. This is what the proposed structure was, because we didn't know. So we based it off of Fret and cross-linking and things like that. So that's why we were talking, Nick, about the fact that it can fold up on top of itself. This is one from Bacillus, but the human is actually very similar to it. But in the lighter color is the pRNA, what we just saw. Does anyone know what this one is? It's another type of RNA. Anyone recognize it? It's a tRNA. Mm-hmm. And this is a little tiny protein. Okay, and this is taken, I cut it out of one of our fingers. And so, yeah, it's a little tiny protein. This is the substrate, by the way. This enzyme, all it does is it cleaves a very specific bond. Because tRNA is just like a lot of the other RNAs, it's made in a precursor form. So it has to be cut first on the 5' end by a very specific bond. Like, it literally matters on the phosphate whether it cuts on the 5' or 3' side. If it cuts on the wrong one, you die. Okay, so it's that precise. And the protein just helps hold it in place. It actually helps it bind the substrate versus the product. In bacteria, in humans, we actually require both the protein and the RNA. Okay, so here's another one. I didn't write down what this one was. RNA quaternary structure. Oh, this is for RNA quaternary structure. Just an example, because this is technically more than one RNA. Does anyone see that? Here's 1 5'. Oh, whoa! That made me seasick. Here's 1 5' end, and see it goes around. That's kind of hard to follow. And there's its 3' end. Here's another one. Here's another big one. So there's are actually technically 3 different RNAs that are coming together to form a complex. That looks beautiful. That's very Pollock-like. Yes.
How do they know that it's separate RNA at the top, but at the bottom it's not?
So I don't remember what paper I took this one out of. If this one's ribosomal or not. What they do, sometimes they're able to crystallize it, so you can tell. What they do otherwise, there are actual programs that are based off of biophysical data, and they can know based off of the structure, and they can propose whether it's going to form helix or not. Because, for example, if we look at this one environment number 2600, this is a tetraloop, and what it does, the computer can do it much faster than we would by hand, but it's called a GNRA tetraloop because it's G, any nucleotide, R, and then the A. And so whenever it sees, the computer recognizes, okay, if it's GNRA sequences like to make loops, and it's proposed right here next to a helix, well, could form a helix around that loop. So that's what they do. So this is a secondary structure map of a primary structure. Because as in 3 dimensions, it's going to look, like that. Okay. Okay, well, we actually already went over that. Yay! So I used to always keep coming back. Now we go over the types of RNA. Whenever I first started teaching biochemistry many years ago, 11 years ago, I've been here 12 years. The first year, Dr. McConkie taught it. Biochemistry this one semester class, Dr. McConkie taught it. It's like, no. This is my field. It's not her field. So I think she was really glad to get rid of it. So she was asking lots of questions, like, what's this and what's that? Okay. But what I was going to say, the very first time I taught it, actual Professor Wagner was in my first biochemistry class I taught, many years ago. But whenever I asked students, I said, so what are the different types of RNA? Usually I would get, what are the types? Well, we've seen some of the types now. And there are more beyond this, by the way. But I would get mRNA, and I'd get tRNA and rRNA, the big 3, so to speak. I'd say the big 3 based off of their fame and not necessarily in their abundance. That's really important to know. Alright, but there's a lot of different RNAs. I picked some of these. They're all the time. In fact, piRNA is really relatively new. I just added it on 3 or 4 years ago, maybe at most, because whenever I first started thinking about chemistry here, I don't think it had been discovered yet. Okay, so, and now they think that it's actually highly abundant. Because whenever I started, and I told this, I don't know if I told it this class, but I told it to pretty sure neurochemistry and definitely other classes. Whenever I first started biochemistry 20 plus many years ago, okay, we were told about the genome. Okay, there are, I don't remember now, 6 billion base pairs of DNA, or 6 or 9, 6 billion, I think, in humans. But like 90 some odd % of it was junk. And so they used to just tell us it was junk DNA. It had no real, value. And now they're finding out that a lot of that so-called junk actually is very important. And now there are actually a lot of diseases that are being associated with these non-coding RNAs, is what they're called. Okay? In fact, that's not even on here. I should have that on there. There's another kind called LNC for non-coding RNA, and another one's called LNC for long non-coding RNA. But like I said, they're actually like the long non-coding RNAs that are helped keep histone's functionality in check. Okay, so let's just start. If we give like a little spiel. But what I was going to say, another thing is nowadays, I think our classes earlier on, how many of you guys know what miRNA is? Does anyone know what miRNA is? It's microRNA. See, now more and more people know about micro and SI. Yes, small interfering technically, but yes, they are. And they technically, it's kind of nitpicky on microRNA versus the small interfering RNA. It's kind of interchangeable. Does anyone know what snRNA is? You probably guess what the S is. It's the same as the other one. Small. It's small nuclear RNA. What about no RNA? What? Small nucleolar RNA, tRNA. No. It's one of my favorites. Another reason why I had to stick it in here. That I'm not going to get to it. It has to do with like the Pee-wee. It's the Pee-wee interacting RNAs. And we'll talk about Pee-wee. Because Pee-wee is. I don't know. I still say that Pee-wee RNAs were discovered by a feminist.
How do you spell Pee-wee?
P-I-W-I. It's like, I want to say it stands for, and I'll look it up in my notes. P, element, interacting, wimpy testes. What was the other I? The W's for wimpy testes. Yeah, in which but notice the T did not make it. It's not Pewdie, it's Pee-wee, because that's why I say that it's probably a feminist that thought. Because what happened was they first noticed that whenever they knocked it out, the result was the Drosophila and mice or whatever would have, the males would have very small testicles. So they call it Pee-wee. So, all right. So what does we're going to start with tRNA. What does tRNA do? The T stands for transfer. What is it transferring? I've asked this question before people. What?
Amino acids.
Amino acids. Okay, it transfers amino acids where? To the ribosome. Next question. What?
P site of the ribosome, right?
Is it going to be the P site? Is it going to be the A site?
Alright,
What? No, so the A stands for amino acylation. So peptide bond formation, and then the empty site is APE. So ape. But all right. What does the structure of tRNA look like? A what?
upside down L.
Thank you. Well, I guess it could be a regular L, right? You know, you only see it this way, right? But all you have to do is sort of, the other way. But yeah, it actually looks like a boomerang or an L or 7. Some people say 7. It does not look like this. This is the way that they usually draw it. And they draw it this way because that way you can clearly see all of the different helices and the loops and all that kind of stuff. I do want you to know the parts to tRNA. Which to me, this is like a little, like a game. It might be sorry, like little games.
Yeah.
Okay. But there are parts to tRNA. Okay, so you need to know the parts to the tRNA. And once again, we go from 5' to 3'. A lot of times they would call this the cloverleaf structure, but it doesn't. In fact, I'll skip ahead. All right. So, but if we go back here, the different parts to it, once again, we have the 5' end and the 3' end. When tRNA is first transcribed, it does not look quite like this. This portion right here, helix P1, technically it's got 2 names. A lot of people call it the acceptor stem, but some of them also call it the amino acyl. Okay, that's the more technical term for it. Why do you suppose they're going to call it the amino acyl stem? What does that name sound like? The amino group? The acyl group? The amino acyl, what is an amino acyl? What? What? It's the amino acid. Like literally, go back to your organic chemistry rules. Amino acid, and they often they add YL. It's amino acid. So yes, you're right. It has an amino group. Yes, it does have an acyl group, but it's amino acid. But yeah, the amino acid actually gets added on right here. Okay, on the 3' end. That's where it's going to be amino acylated. And so that's why many times it's called the amino acyl stem. It's also called the acceptor stem because it accepts the amino acid there. Okay. All right, this one right here is called the D loop, and this is called the D stem. And the D stands for the fact that they have to have a modified U here. It's a dihydrouracil, okay? So it's a modified U in that loop. The letters that are there, that they have left there, are ones that are conserved, highly conserved throughout all species and all tRNAs. The next stem, and they always name the loops after the stem. So the D loop is on the D stem, which is for the dihydrouracil. The anticodon loop is on the end of the anticodon stem. Okay, and so therefore, what do you suppose is down here? The anticodon. Okay. This loop right here is called variable because on some of them it just looks like a little bulge, and on others, I think it's one of the tRNA phenylalanines, if I remember right. It's huge. It looks like a big long arm that sticks out. That's the one part that varies the most. Well, it varies from tRNA. This one right here, they call it the T stem or the T C stem because this is the T loop or the T C loop, and that's because every tRNA in that order has to have a T, this pseudouridine, and then the C. Okay, and then we're back up to the amino acyl stem. That's what it looks like in 3 dimensions. That's why it looks like a 7, an L, an upside down L, a gamma, if you want to look at a capital gamma from Greek, whatever flows. The state of Florida, you know, if you flip it over. Good one. Kudos. So that's why this is just to show you it's color coded and to show you where the different colors, like how they line up to make the gun. You can also think of it as a gun. Pow. You better not big enough. But yes. And one other thing I wanted to point out is that the anticodon loop right here, the anticodon itself, even though it's always drawn what's on the bottom, it's not really on the bottom. Like, it's kind of cockeyed. Does anyone know why from genetics? What's so special about that one position? The one that on this one is up high. Are all of the, what is the anticodon? You know, tell me it's the antithesis of the codon.
It's the mirror of the sequence on the RNA.
The mirror of the sequence of the RNA. Mirror? Not mirror, but the complement. Mm-hmm. The complement of the sequence of what RNA? The mRNA. The mRNA has a codon. How many nucleotides are in the codon? 3. The anticodon would have how many nucleotides? 3. What about that third one? What's so special about that third position? It's the wobble. And so now you can kind of see where 2 of them are going to connect really well on the mRNA. That third one's going to wobble. Why do you have the wobble?
Degeneracy.
The what?
Degeneracy.
Yes. Oh, that's like a good word.
Oh, we just learned it in
Oh, good. I was going to be so impressed if you remembered that from March ago. Yeah, you're like, you should have just went with it. Of course I know that. Okay. So, as I mentioned before, this is the way tRNA is made. This is a universal pathway. Okay? So, bacteria do it, humans do it, yeast do it, mitochondria do it, the birds do it, the bees do it. So, all right. First, you make what's called pre-tRNA for precursor tRNA. So this is pre-tRNA. There's a 5' leader. There's an extra part on the 5' end, and there's an extra part on the 3' end called the 3' trailer. And in some species, like eukaryotes, they have this extra part. Which one's that more commonly called, that extra part that has to be cut out? The introns. Mm-hmm. So that's got an intron, which stands for intravening sequence, by the way. It's just, right. All right, so the very first step, and it's the concomitant step. Does anyone know what that means? Concomitant. I can't remember if there may be two M's in that, concomitant. It's the one that's the committed step to the pathway. Okay. It just doesn't look right. It may have, like I said, it may have 2 M's. But just in case I call it that, your book would never say that. All right, but it's a step that's kind of like the rate, it is the rate limiting step of the maturation pathway. It's one that once that enzyme works, and it's called the ribonuclease P, then it's going to go forward. Okay, so this is the part that gets highly regulated. And all it does is it literally cuts at a very specific bond. It's got to be on the correct side of the phosphate even. Okay, remember there's a phosphate. So it's got to be on the correct side of the phosphorus, not even the phosphate, but the phosphorus atom. If it cleaved on the incorrect side, you can die. Remember, most of the nucleotides, most RNAs form those 2' 3' cyclic phosphates. RNase P, when it does tRNA, does not do that. It actually will form an alcohol and a true phosphate. Okay. Alrighty. So, yeah. So it's got to be on the correct side of the phosphorus. Then the second step is the trailer gets removed. And this one shows a 3' endonuclease, but there are also other ones that they sometimes call 3' tRNases, which are exonucleases, which just help chew it up. So sometimes that 3' trailer is really big. Then, if the organism doesn't have that CCA on the 3' end already in the gene, like a lot of ours, we already have it included in our gene, then it has to be added. And the enzyme that does this is called tRNA nucleotide transferase. It transfers nucleotides to tRNA. And it literally has to be a CC and an A, because that A is what gets amino acylated. If you have a different sequence there, you die. Life is precious.
So, since we already have that, whatever.
Then most of our genes, I should say most of our genes, I don't know if all of our genes, most of them have it in there. But for the tRNA, because we have a lot of tRNA genes, there's not just one, we have a lot more, it's like what? Close to, I can't remember now, 60, give or take. But what I was going to say is, and then the last step, but if it's not in there, then the tRNA nucleotide transfers will have the CCA into it. And to it. Then the final step is the exon. If the exon is there, if the intron is there, it gets excised. And so, all right. And so now we have the mature tRNA, which can be amino acylated to go on to do the work. And so it's involved in protein synthesis in a couple minutes. Translation. Make sure you know its role. So that one shows you the phenylalanine that's added to it. See, the amino acid is actually quite small compared to everything else. It's very important. One that gets overlooked is it discerns the sequence proficiency. And you may have already covered this in microbiology. It's really important. Different species have different numbers and the high proficiency of genes for tRNAs. So you have that universal code that you probably have seen in every genetics and biology book where they got like AUGC on one side by this, and then you just look. But what happens is, let's say if there were 4 leucine genes, it may be that humans predominantly use one codon, whereas E. coli predominantly uses the other, and it's based off of how many tRNAs that we have for it. Okay. And so it helps discern the sequence proficiency. It reads the codon by its anti-codon. And once again, there are many types. What about the? Oh, why do I have that on there? Yeah. What about the stop codon? What does it code for? Are there tRNAs that have the code for the stop codon? Yes or no? Yes. But they don't carry an amino acid. What do they carry? Does anyone know? This is actually really good from organic chemistry. Like if I was teaching organic chemistry at the time, I'd tie it into the kind of reaction that's occurring, which is an amide bond formation. If you want to break it off, there's a molecule that we have a lot of that's a great nucleophile, and that's water. And so technically they code for water. They carry a water molecule there to kick off a protein. So, yeah.
So stop codons.
They code for water.
Mm-hmm. Code for water, just interaction with water.
No, where that phenylalanine would be, there'd be a water. That's why they see a code for water, and they call it stop codon, because it's going to stop the translation by having the water act as a nucleophile to form the C terminus of the protein. Because when you got at the end of the carboxylic acid, if you kept going with your amino acids and stuff, you're going to keep forming amides instead of carboxylic acid. Okay, so at this point, I'm going to go ahead and stop.